Let's start part 2 of this lecture, which is to go over how GeneMANIA works. So in a very short summary, here are some keywords defining GeneMANIA. GeneMANIA is a functional interaction network. Nodes in the network are genes and they are connected by edges if they are functionally associated. For example, the proteins may be known to physically interact or to belong to the same pathway or to co-localize. All functional networks are merged into one global network, and there are weights associated with them. To predict the function of a gene, GeneMANIA uses the concept of guilt by association, and GeneMANIA finds genes and networks associated with our gene of interest. Finally, GeneMANIA is available as an interactive web app and a Cytoscape app, and I will later make some suggestions on when it's easier to use the web app or when it's better to use the Cytoscape app. So we have a first question. What does my gene do? In this case, we have two elements. So we have the source of the functional interaction networks, and the second element is the query list. Here it's one list. So the gene recommender system is going to look at the functional networks and retrieve by default 20 genes that are related to the query list. Then the functional interaction scores are going to be distributed across the network using by the guilt by association algorithm, and then we can determine the function. Then we have the second question. Give me more genes like this. In this case, we still have the same source of functional interaction networks, and then the second element is a gene list. So in this case, it is context dependent. So the gene recommender system is going to first look at the genes in the gene list and see how they are connected to each other using the functional interaction networks. And based on that, is going to add the other 20 genes to the query list. Then it uses the guilt by association algorithm to distribute the scores across the networks and then determine the function. GeneMANIA uses a various source of functional network, and they are summarized into main categories that we can see here: co-expression, co-localization, genetic interaction, pathway, physical interactions, predicted, shared protein domain. For example, co-expression comes from GEO, genetic interaction from BioGrid, physical interaction from RF, index predicted for I2D, and shared protein domain from Interpro. There are also some attributes that can be drug targets, microRNA targets, transcription factor targets, and a few more. Here is the complete list of these categories, and you can find more information on the GeneMANIA help page for the definition of these networks. So this is the input menu option of GeneMANIA, and you can recognize now the main categories. So you see for each of the categories, you have two numbers. The first number is the number of selected networks, and the second number is the total of networks available. So by default, GeneMANIA has a set of networks that it will use, but you also can choose your own networks if you wish. And if you click on a tab, then you have a detailed source of information to select the networks. Once the network has been created, so once the final composite network is created, you also have access to the networks that have been used for these final networks. And you can even click and expand the tab to trace back the origin of the functional data. So GeneMANIA, so this is an image of the GeneMANIA app, and then you can see the GeneMANIA network in the middle of the window, and then on the left, you can see the networks that you can use the default, or you can choose to do your own selection. And then on the right, you have the information about the final networks and how these networks have been used. So we have seen that GeneMANIA use some context-dependent networks. So it means that they have some weights associated with the networks, the way they have been used. And so this is the final composite networks, and then we can see expressed as a percentage the amount of which category and how they have been used in the networks. So tools for network weighting. So there will be more weight for relevant network. So for example, of the genes in the query list more often connected to one other than the other genes, and there will be less weight if these networks are redundant. So by default, there is a default weighting method, and this default weighting method has the goal to do the function prediction. So it is Go dependent, meaning that we'll have the genes in the query list, and we will look if these genes in the query list are connected using a pathways, and then so all the networks that are connecting the genes from the same pathway will have more weight. So this is for the function prediction, but there are many other options for the weight. For example, equal by network and equal by data type, where the network or the data type will get the same weight. So in summary, there is the weight of the different networks are used to measure the node connection, and there are different options. So automatic for using the pathways, but you also can change to equal by network if you don't want really to use the gene function prediction, but you want to get other information about the network. And then you can see for the same query, we're using the percentage of the result networks that it influence slightly the results. So you can do your own choice. And so GeneMANIA is using guilt by association to distribute the functional interaction score to the network. And so this is how it works. So this is the network. So we have here in red 4 nodes with a high interaction score. These nodes have like a known function, and we see here a gradient of color from white, yellow to red. So red for this nodes with high interaction score. And what label propagation is going to do is like diffusing heat through the network. So it's going to diffuse the score through the network to the nodes that are directly connected to those red nodes, but also to the nodes that are connected to the neighbor nodes. So it's very different from the direct interaction, where here the score are just propagated to the direct neighbors. So label propagation score will propagate direct neighbor score, allowing indirect links to impact the score. So this is an example of label propagation. So this is the 4 nodes with a very high score with a known function. And then after the label propagation, then we distribute the score to these highly interconnected nodes. So these genes that are highly interconnected by the functional networks are probably sharing in a same function. So it makes sense to distribute the score to these nodes. So this is a summary of the GeneMANIA concept here on this slide. So we have all the networks. So now we know that we attribute different weights to these networks to find the composite network, which is the final network. And then some nodes we will have a high functional interaction score, but the nodes that are closely related to these other nodes by label propagation will also have been attributed also with dysfunctional interaction scores, and then we can predict the gene function. So this is the output of the GeneMANIA. So it's a final network. We see the networks on the right, But what we also can do is to color the genes by the function. So if we click on this tab, then we will see some pathway enrichment results based on all genes in this network. And then you can see here the top three functions have been annotated by some different colors. So I've shown you the web app, but there is also a Cytoscape app that is also available and has all the GeneMANIA web app functionalities. And there are actually two ways to launch a GeneMANIA query in Cytoscape. So the first one is from the network tab, and it's basically going to connect to the GeneMANIA web app. But there is also a way to go from the menu apps, GeneMANIA local search. And this one is going to use the data that you've installed in your local computer. And so it's better to use this local search when you have more than 100 genes, because it's going to be more rapid to get the answer. So GeneMANIA uses and recognizes multiple gene identifiers like interest gene ID, gene symbol, or ensemble IDs, and also works on several model organisms. So what have we learned in this part 2? We have learned that GeneMANIA is built to answer 2 questions about gene function. What does my gene do? And give me more genes like this. And it uses functional interaction networks and a guilt by association label propagation algorithm to predict gene function. It uses a network weighting system to answer queries in a context-dependent manner.